Thanks, Matt. And on behalf of the Provost's Office at the University of Louisville, we want to once again thank NIEHS and all of you for attending this outstanding symposium that has occurred over these 2 days on liver and environmental chemicals. Our first talk in this session on chemical hepatocarcinogenesis is going to be given by Tetyana Kobets, and it's in collaboration with her colleague Garry Williams at New York Medical College. I would just like to comment that Garry Williams has been a past recipient of providing the William Waddell Memorial Lecture here at the University of Louisville, and we will have another one in our last session by last talk by James Swenberg. And you may see some additional people coming in during this session to attend that last William Waddell Memorial Lecture. So our first talk will be given by Tetyana Kobets, Xenobiotics and liver cancer.
Well, thank you very much for the introduction. It's a great pleasure to be here. It's a great symposium, great reception, great talks. And I would like to contribute also a little bit to the symposium. And today I would like to talk about xenobiotics and liver cancer. I guess no need to remind that liver cancer is a very complex disease which is associated with a wide variety of risk factors. Among them, infections, chemicals, radiochemicals, of course, metabolic disorders, and some genetic conditions. But today I will talk only about one group, don't worry. It's going to be chemicals. We know that liver is very prone to the injury induced by chemicals because it's, of course, as already mentioned, it is the main organ for xenobiotics metabolism. And also it is a first pass organ for a lot of chemicals. So after absorption, the first pass effects happens when the concentration of absorbed drugs is way lower in the systemic circulation after being absorbed in the liver. So all carcinogens have different mode of action, and of course that influences their genotoxic potential a lot. And in order for us to classify those carcinogens for the risk assessment, they are divided into two big groups, genotoxic and non-genotoxic carcinogens. The genotoxic carcinogens imply chemicals that are capable of direct DNA damage or formation of DNA adducts. And usually their effect is irreversible. And among those chemicals are already mentioned in the course of those 2 days, mucotoxins, acetaminophen, vinyl derivatives, and alkoxy-substituted allyl benzenes, and many others. The non-genotoxic carcinogens are not capable of causing direct DNA damage, but they have some secondary mechanisms that are involved in the carcinogenicity. And under this question mark can be various events like epigenetic alterations that are changing gene expression, induction of apoptosis and promotion of proliferation, oxidative stress, and many others. Unfortunately, we are still not aware of all the mechanisms by which the non-genotoxic carcinogens are involved in the process of carcinogenesis, and that's why it is hard to make a more precise classification of the chemicals at this day. Of course, I won't talk about all of those chemicals, that's too much of the talk, but I will focus today on two groups. The first group will be alkoxy-substituted allyl benzenes, and the second one would be furan. So when do these compounds get in action in the process of carcinogenesis? Well, two basic phases, neoplastic initiation and neoplastic development. The carcinogen or its reactive metabolites through direct or indirect DNA damage is causing the initiation of cell, which through the clonal expansion in the promotion phase is turned to pre-neoplastic lesion and later to neoplastic lesion. And in the progression phase, it's turned into malignant neoplasm with growth advantages. As I promised, I'm going to talk today about specifically about one of the groups of the carcinogens. It's alkoxy-substituted allylbenzanes, flavored agents that are present in our diet in cinnamon, anise, basil, and other spices. And here are some representative from this group: safrole, estragol, methyleugenol, and anethol. Importantly, those chemicals show to be cytotoxic and hepatocarcinogenic in mice. And here are the results of some bioassays provided in rodents. And safrole, estragol, and methyleugenol resulted in the development of hepatocellular adenomas, carcinomas, and cholangiosarcomas. And due to those results, they were classified as possible human carcinogen, group 2B by IARC, and reasonably anticipated to be a human carcinogen by NTP. All those compounds are sharing the similarity in their chemical structure, and it is allylbenzene nucleus, the alkoxyrin substituent, and also the presence of double bond in the alkyl side chain. Now, that might be a really small peculiarity of chemical structure, but there is a hypothesis that the position of this double bond is highly involved in the potential of the chemicals of this group to involve carcinogenesis. Thus, as I already mentioned, the compounds with a double bond on the proximal side are genotoxic in rodents. At the same time, the chemical analogous with a shifted double bond did not share this genotoxic potential. So most probably, the different metabolic roads are causing these differences. So in order to study the dose-dependent effects of methyleugenol, the group in New York Medical College provided this study. And in particular, the male F344 rats were fed through gavage different doses of methyleugenol for up to 16 weeks, and it was an initiation phase. And after the gavage with methyleugenol was discontinued, rats were maintained on the either control diet or diet that contains 500 particulometer of phenobarbital at so-called promotion phase. In the end of the study, livers were excised, and the endpoints of the study were the detection of DNA adduct formation using the P32 post-labeling assay, and also the indication of incidences of preneoplastic and neoplastic lesions in livers as indicated by immunohistochemistry. So this is the results of their study. Methyleugenol, already in initiation phase, was capable of formation of DNA adducts, and interestingly enough, there was, as you can notice, the dose-dependent pattern of those adducts formation. And after 16 weeks of diet, the formation of DNA adducts was numerically higher than at 8 weeks, so probably there is some cumulative effect is happening. 40 weeks, that means 24 weeks after the stop exposure to methyleugenol, the levels of DNA adducts are already way lower, probably due to reparative processes, but still the dose-dependent manner is still saved. The formation of hepatocellular altered foci, which are the preneoplastic lesion leading to the neoplasms in future, already happened also in initiation phase on the high dose, and was observed during the whole, pretty much the whole exposure period. And in the promotion phase, in both high dose and mid dose, it led to the formation of hepatocellular altered foci. Well, expectedly, the promotion with phenobarbital would cause more formation of more tumors, but what we noticed was that the multiplicity of those tumors in the rats that were fed with phenobarbital was higher than in control group. So methyleugenol was acting in the initiation phase, but phenobarbital was enhancing the effect of methyleugenol. Of course, animal models contributed a lot to our understanding of chemicals' mode of action and carcinogenesis in particular, but unfortunately they're very cost-efficient and time-consuming, and not even mentioning that they are not very relevant to humans. So of course there is a need to develop new models, and I would like to introduce you to several new approaches to the studying of carcinogenic potential, and in particular I would like to talk about in ovo carcinogenicity assay. That's the carcinogenicity assay that investigates the potential of the compound to produce preneoplastic lesions in turkey fetal livers in ovo. And if you would like to learn more, here are the number of publications on this subject. And the same in ovo approach was used for the chicken and turkey egg genotoxicity assay. And in those assays, we investigate the potential of the compound to produce DNA damage, and in particular DNA adduct formation and DNA strand break in the livers after these chemicals are injected in the eggs. So here I would like you to show you some examples from the chicken egg genotoxicity assay that we saw in after the exposure of the chicken eggs to acetylaminofluorine. And as you can see from the results of nucleotide postlabeling assay and the Kobets assay, indeed this known hepatocarcinogen was capable of forming DNA adducts and DNA strand break, and also to bind those genotoxic effects with the phenotypic changes. We did the analysis of liver section stained with hematoxylin and eosine. And control, used for control coliform is the derivative of oil, so it caused a little bit vacuolization of liver, but none of the big changes. However, the injections with 2-acetylaminofluorine caused the hepatocyte hypertrophy, and most importantly, in the cholangiocytes, we noticed already anisocytosis and anisokaryosis, which are the markers of neoplasia already. So that was very interesting studies, and nobody showed it previously in the eggs. And no inflammation was noticed, and yesterday from the talks we also saw that aseptic mice did not develop any inflammation, and our embryos are also aseptic since they are still in the egg. So we decided to use the turkey egg genotoxicity model to assess the genotoxic potential of some of the alkoxy-substituted allylbenzane. I must also notice that this experiment is still in progress, so more chemicals are to be assessed. But so far we have results for chemicals such as safrole, estragol, methyl-eugenol, and anethol, and the doses we chose were based on the lethal dose 50 for rats. So all those chemicals, different doses, they were injected in the medium white turkey eggs on the day 22, 23, and 24 of their incubation. The total incubation period for turkeys are 28 days. And then we opened 3 hours after the last injection. We opened the eggs, we checked the viability of the embryos there, and we excised liver. And the endpoints for our studies were DNA adduct formation and DNA strand breaks. So here are the results of our study. Very congruent with the literature already published. Safrole, the first carcinogen from this group that was actually found to be carcinogen, caused the linear dependent formation of DNA adducts with a low viability of eggs on the high doses. The same with methyl-eugenol. Also, the DNA adduct formation was up, but in a dose-dependent manner, and caused also the lower levels of viability. And I must come back to the results of in vivo study with rats, that we also saw the formation of DNA adducts. And most interestingly, I didn't show this data here, but the pattern of DNA adducts was also the same in the eggs and in the rats. The estragol caused the formation of DNA strand break, and in higher doses also the formation of DNA adducts. So all those 3 compounds, congruently with data already published, show genotoxic effect. The anethol, I want to bring back the chemical structure. It's the only alkoxyallylbenzanes with a shifted double bond. Did not produce such a big genotoxic effects, which is congruent with the data, with the lack of the carcinogenicity of this compound, which we found very exciting. So these results also prove that indeed, estragol, methylugenol, and safrole was capable of causing direct DNA damage, whereas the anethol did not produce any genotoxic results. So probably the position of double bond on this alkyl side chain play an important role in the genotoxicity of the compound from this group. Well, the word epigenetics sounded a couple of times already during this presentation yesterday and today, and of course those are another important events in the response to the chemical exposure, and those are heritable changes in gene expression that are not related to the changes of DNA sequencing, and those changes include DNA methylation, histone modification, expression of non-coding RNA. And furan, one of the another toxicant that we are exposed to through the diet, through the manufacturing, since it's in heat-treated products, it's used in the industry, and most concerning it's in baby food. And as shown by NTP study, the exposure of rats and mice to furan caused the formation of cholangiocarcinoma, hepatocellular adenomas and carcinomas, and monocellular cell leukemia. And also it has been classified as a possible human carcinogen by IARC and reasonably anticipated to be a human carcinogen by NTP. Metabolic activation of furan is possibly involved in the toxicity of this compound, since in liver, under the influence of CYP2E1, it is forming the very reactive metabolite, which is capable of binding to DNA and proteins and causing toxicity and carcinogenicity effects. However, it is still not clear if all of these effects are involved in the hepatocarcinogenicity or there is something more. So during my postdoctoral fellowship in the National Center for Toxicological Research, we were interested in if the furan would be capable of producing some epigenetic alterations after being exposed. So with this in mind, the male Fisher 344 rats were, through gavage, they're fed with different doses of furan. So the dose 0.92 and 2 milligram per kilogram by the NTP was found to be non-carcinogenic, whereas the dose 4.4 milligram was already shown to be carcinogenic. And our poor rats were fed for 90, 180, and 360 days, and then the livers were excised. And here I need to notice that even in the carcinogenic, even after exposure to carcinogenic dose, no morphological changes were observed. We were very surprised with this, actually, with this finding. But we're still interested, maybe still epigenetics is happening there. So what we found, global DNA methylation level, one that is usually in human genome, 90% of our DNA is hypermethylated. So if hypomethylation is causing the activation of some tumor suppressor genes, probably the repetitive elements, of course, that are resulting in the higher mutation levels. So what we noticed was the dose-dependent decrease in the DNA methylation, and especially in the higher dose. And most probably, this decrease in DNA methylation was caused by the decrease in the levels of de novo DNMT transferases, 3A and 3B. Those are those that bring in new cytosines to the DNA, not the DNMT3 that is the DNMT1, but is methylated already, methylated DNA. We also were interested in the histone modifications. They are very involved in the genomic stability, and also histone 3K56 in the literature is known for its involvement in DNA reparation processes. And what we noticed was also after a year of exposure, again, the decrease in histone trimethylation on lysine 9, lysine 20. The same happened with acetylation of histones. So probably they are not capable of protecting DNA anymore. So something is happening that allows them to open access, more access to DNA. And our findings, they're also proved after we analyzed the expression of histone-modifying genes that are those, the histone methylation genes and histone acetylation genes. They were also, their expression was reduced at the higher dose after the year of exposure. And also the promoter methylation. We already heard that if methylator is promoted, there are a lot of methyl-cytosine, so all its active sites are blocked, so it's probably not capable of, not active. And we were interested if we will see the same thing after the exposure to furan, and we looked specifically at ROS1A, one of the tumor suppressor gene in the humans and in the rodents. And what we saw was that its promoter region was hypermethylated, especially 1 year after exposure on the higher doses. However, its expression was downregulated. So probably there is a connection there. Well, methylation goes up, but expression goes down. So to sum up our findings, the exposure to furan, even in the absence of morphological changes, did cause some epigenetic alterations that can be the biomarkers of exposure. And those alterations included global DNA and gene-specific changes in global DNA and specific gene methylation, changes in histone lysine methylation and acetylation, altered expression of chromatin-modifying genes, and altered expression of tumor suppressor genes. And actually, the absence of those evident morphological neoplastic abnormalities allowed us to conclude that those changes were probably due to the exposure to furan, but not were caused by the malformation. And also, these findings are very different from the methyluginol. If you would recall, the methyluginol was already active in the initiation phase, causing the changes on the 8 weeks, whereas here, still it took furan a while to produce any changes. So that is also the difference in the time of the different carcinogens. It's also very important to remember. So I would like to acknowledge the great group in the Chemical Safety Laboratory of the Department of Pathology of New York Medical College, Division of Biochemical Toxicology, National Center for Toxicological Research, where I did my postdoctoral fellowship under the sponsorship of Oak Ridge Institute for Science and Education, and of course for National Institute of Environmental Health Sciences for sponsoring and supporting our work on furan. And of course, thank you very much for joining me for this talk today. And with this, I'll take your questions.
Thank you very much, Tetyana. Wonderful talk. We have time for a few questions.
Very interesting. On the turkey egg assay,
yes.
Have you or anyone looked at what P450s and other biotransformation enzymes are actually expressed in the embryos at that stage of development?
We did not look specifically on P450, but we know that the metabolic pathways in those livers are very similar to what we see in mice and in humans. And interestingly enough, all those, we were more interested in the phase 2 of metabolism because it's the main detox pathways and the main pathways of toxicity in our cases for our chemicals. But it is, of course, it's an interesting thing to look in our future studies, but from what we know, the pathway of metabolism are very similar.
May I add to that? Because you haven't seen these data, but we did a collaboration with the drug metabolism group at Wyatt. They had a system for measuring the activity of 5 major human CYPs by simply doing mass spec on the expected metabolites. And the turkey is expressing 4 of the 5 major human CYPs.
Yes.
One really enjoyed the presentation. So one question: you show when you had your histopathology, you show what I believe was proliferation of cholangiocytes, correct?
It was actually the anisochriosis. Well, yeah.
So the question is, when you look at DNA adducts in liver, have you looked at different cell populations, for example, hepatocytes versus cholangiocytes, and determine whether there might be a differential level of DNA adducts between those 2 cell populations?
Well, we did not look at that yet, but definitely that's an interesting thing to look at. We basically just took the liver and in the whole liver, so we probably should do this separately for the cholangiocytes as well. But also together, we're more thinking that the that the changes in the hepatocytes are inducing all the promotion of cancer. So we're more,
Because I believe that Garry was saying earlier that those might be the sources of some forms of
oval cells,
cholangiocarcinoma,
Yeah.
right?
yeah.
By the way, we're just getting started.
Yeah, it's just the preliminary data. So.
Tetyana has been brought in to do gene expression work in this model.
Yeah, so we want to do the phenotypic anchoring to the gene expression.
One final question.
Yes, so the question was, if I understood it correctly, that there was a delayed effect in the
Furan, yes.
So what's the implication in terms of mechanism?
Well, it means that, well, if we'll go back to the, I'm sorry, if we'll go back to the, okay, I'm definitely moving the wrong direction. Okay, so here's our hepatocarcinogenesis model. So the implication of this is that epigenetic effects can be prolonged, can happen in every stage, and it requires a lot of exposure to it, and we are all exposed constantly to those chemicals. And if it keeps accumulating in our body, even if first even for a year it didn't show any changes, it doesn't mean that nothing is happening. There is still a lot of happening, and it's still causing a lot of adverse effects.
Bob, did you want to have one quick question? Or not?
I just wondered, do you always use oil for your vehicles? I'm just wondering whether the steatosis you see is actually...
Well, actually, for our model, well, it depends, of course, of the chemical that we use. And the vehicle that we use, it's either water or the colifor. But already I know that a lot of vehicles were previously tested in the eggs, and those two, the water and the colifor, were shown to be causing the less some cytotoxic effect in the eggs. So that's why we're using those two vehicles.
Thank you very much.
Thank you very much.
Okay, our next presenter.